Thank you, Tad. Welcome.
Nice. Thank you very much for the introduction. And so I'd like to start by first outlining some of my motivations for coming here from Helsinki. The first of which being that Spain is slightly more southern and therefore slightly warmer. So this was taken a week before coming here, and it was about negative 30 degrees. And you can walk on that frozen river there, or snowmobile if you so choose. But more seriously, sort of why I came here is in Helsinki. I'm doing a lot of work on applying ecological niche models to understand macroecological patterns. But starting maybe about 6 or 7 years ago, I had a really strong interest in the structure of ecological networks, and specifically host-parasite networks. And so this was sort of an excuse to get back toward this area of research. And so in a simple sense, this host-parasite networks can be represented by different colored circles, where we have hosts as red circles and parasites as blue circles, and the interactions between them being instances of a parasite parasitizing a host. And this is where a lot of my work sort of started, and where a lot of it actually continues to go. But what I'd like to talk about now, or recent projects, looking at more the global context of this. And so a lot of times these networks are formed based on the interactions between hosts and parasites in a single location. And we analyze this and we say something about it. But that doesn't allow us to look at large-scale spatial or environmental gradients that could structure these networks. And so what I'd like to sort of shift more to is looking at networks that are connected to other networks across space. And a lot of this thinking that I have been doing sort of stems from my interactions with a group of scientists in the United States that started during sort of the middle of my PhD, and so it's 5 or 6 years ago. And this was the Macroecology of Infectious Disease, RCN, which stands for Research Coordination Network. The idea was you bring together a group of macroecologists with a group of disease ecologists and see what comes out of it. Try to bridge the two disciplines, basically. And one of the first sort of outputs from this effort was this review paper that was really great. It set up a sort of roadmap as far as what were the conceptual and theoretical relationships between macroecology and disease ecology, and what were the big open questions that still had to be yet be answered. And a lot of them dealt with sort of like classic macroecological scaling rules, things like body size distributions, latitudinal diversity gradients. But I saw this and I said, man, what would be cool is now we can actually look at macroecological scale patterns of networks, of host-parasite interaction networks. And as soon as I had this thought, I started digging around and searching for papers. And I'm sure many of us have been in a situation before where we find it's already been done, in a sense. And so people had, for a while, examined macroecological patterns in plant pollinator networks or consumer resource networks. So there's a few studies on that, but not much on host-parasite networks. And I think largely this had to do with the sort of paucity of data. So there wasn't that much data on host-parasite interaction networks distributed at large spatial scales. But I was lucky enough to find such data. And so the first year of the RCN, of this group, basically was about searching for data. And so I sort of struck gold here. And so these are the data that will form the rest of this talk, as well as several ongoing projects. And they're data on host-helminth interactions, and so worm parasites, things like trematodes, cestodes, nematodes, acanthocephalans. And they're compiled by the London Natural History Museum from a large set of museum records and published studies. They're georeferenced to the geopolitical scale, which is not ideal, but also including lakes and coasts and islands, which is really interesting. And so part of my work early on was to develop an R package to access and clean this data resource. And that's I called Helminth R, and I think this is the only thing resembling a parasite that I will show you. And so no disgusting parasite pictures in this talk. And so this package is supported by R OpenSci, which is a sort of open science collective of R developers that make and promote programs or R packages for the acquisition and manipulation of freely available data. And so I encourage everyone to check it out if you have any interest in Helminth parasites, and feel free to ask me any questions about that. And despite the sort of like limited taxonomic scope, right? This isn't all parasites, so it's no like ticks or any of the cool stuff like that. This is just helminths. We see that it's actually currently the largest host parasite database to date. And so on the x-axis, we have the number of unique host species in the data. On the y-axis, we have the number of unique parasite species. And then the point size is proportional to the overall number of records in the database. And you see things like the Warday database or like even the global mammal parasite database actually pale in comparison on this log-log scale to data just on helminths, which I think is really cool. And so using these data, we can start to build networks at each locality, at each of these geopolitical locations. And so these are things like countries, states in the US, lakes, as well as countries that don't exist anymore. And so the Soviet Union is still sort of listed in there, and there's records that have to be cleaned out before the data can be used. However, this got me back to thinking, hey, like now it is possible to look at macroecological patterns in host parasite, and specifically host-helminth interaction networks. And so what I'd like to talk about today are sort of two projects that utilize these data to look at community structure and species roles in ecological networks across space. And so the first question we'll look at, how do host and parasite communities and networks change across space? And the second question that I'll address is the conservation of species roles across different networks. And so to address the first question, it's sort of a truism at this point that hosts and parasites are distributed non-randomly in space. And so this is a map of standardized parasite species richness, which is parasite species richness divided by host richness. And we see a lot of spatial variation in this. But this doesn't tell us about the actual community composition of hosts and parasites in each location. But since the work sort of before Grinnell, before Clements, before MacArthur, there was this idea or this belief that community composition decayed with ecological distance. Communities that were farther apart in space tended to be more dissimilar, also called Tobler's Law, I believe. And this has a ton of evidence. It's actually like pretty easy to find evidence for this. And so these are two review papers that basically said like, yeah, this is a thing. And part of the reason why there's been a lot of papers on it and why it's such a big thing is that the dissimilarity of two communities is basically beta diversity, which has always been like a hot topic and always been something that ecologists have studied. And so the idea is that if you have a gradient of distance, here on the sort of columns represent distance, and you have communities where species are occupying rows here, we can go and we see a community change over time. So we start with this green community here. We go through time, and by the end, at the farthest distance, it shares no species in common. It is completely dissimilar from the community that it started from at some distance. And it's sort of easy to think about what community or what a distance decay relationship would mean for host communities. It would mean that there's spatial or environmental gradients that are controlling, or dispersal limitation, that are controlling the composition of communities. But then when you think about parasite communities, it changes interpretation somewhat, because parasite communities are not only structured by environmental and spatial gradients, but they're also structured by the availability of suitable hosts. And so we have situations where if we see a distance decay relationship in parasites, it could be a function of spatial or environmental gradients, or it could just be a function of the host community. So an entirely neutral parasite assemblage could form a distance decay relationship if the host communities had a distance decay relationship. So it could just be an emergent property of the host communities. And so I started to sort of try to disentangle this using these host-helminth parasite data. And so this is a map of sort of sampled locations that we looked at. I think there was about 300 sampled locations, and how we measured. So this is a pairwise measure. And so every single pair of these sites is compared in terms of host community and parasite community dissimilarity. And we use an index called Sorensen dissimilarity. And basically, in green, you have the number of overlapping species, the number of shared species between the community, or between the two communities. And in orange, you have the total number of species in both communities. And so this is dissimilarity, which means that a value of 0 actually corresponds to a completely similar community, or a pair of communities, same species. And a value of 1 would indicate complete dissimilarity. These guys share no species. And so using these data on host and helminths, we can start to look at this sort of scaling between geographic distance between sites here on the x-axis and community dissimilarity on the y-axis, where on the left panel we have the host community dissimilarity, and on the right panel we have the parasite community dissimilarity. And what we see is that communities become quickly dissimilar. So within 4,000, 5,000 kilometers, we see communities become almost entirely dissimilar from one another. And this sort of makes intuitive sense why we wouldn't think that the parasite community in China is the same as the parasite community in like Mexico, like somewhere very sort of more distantly related. But we can narrow down and look at like a smaller scale. And so what I did is I subset it to just states in the United States of America and looked for this same distance decay relationship, and found strong evidence that this is still a thing at smaller spatial scales. And then I sort of related host community dissimilarity on the x-axis to the parasite, to the helminth community dissimilarity on the y-axis. And you see a really strong, like nearly like one-to-one relationship, which this is sort of speculation, but it seems like this would be indicative of either host communities and parasite communities following the exact same environmental or spatial gradients, which doesn't seem that likely, or that the host community dissimilarity is driving the parasite community dissimilarity. But then, when I started this talk, I said, I'm going to examine macroecology of interaction networks. And all I've done is look at communities. And so what I wanted to do next is look at the actual structure of the ecological networks and take two networks and be able to compare them. And this is sort of a difficult thing to do, as these networks differ in the number of interactions they have, the number of species they have. And so how do we really go about comparing two networks of different sizes and different fills? And so I used this, I won't go into too much detail, but I used a metric developed by Schieber et al. In 2017 that has pretty good performance on simulated and empirical networks, but it was designed originally for unipartite networks. So what we have are bipartite networks in that they have two classes. We have host and parasite, or plant or pollinator. And so what I had to do was reduce it down and do something called project the bipartite network into basically two unipartite networks in order to use this measure. And what that means is I develop networks of basically parasite sharing on the top or host sharing on the bottom, where In the parasite sharing network, nodes are host species that are linked together by common instances of parasitism. And so what we're looking at in terms of the dissimilarity of networks over time are these two series of networks. And that produces plots very similar to what we sort of saw before, except way uglier. And so we don't see the classic distance decay relationship when comparing these two networks, or the host sharing network and the parasite sharing networks. And so what this means to me is that we have host and parasite communities that decay, that become increasingly dissimilar very quickly across geographic space. But the networks don't. So that means that functionally, or different species in these networks are playing functionally similar roles in the structure of the networks. And so that got me thinking. And this is sort of a project that I'm working on here. And it's, does the same species perform a similar role in different networks? And so host and parasite species, despite the sort of rapid decay that we saw in community structure, host and parasite species are in multiple networks. And so this is an example of the distribution of one species, which I'm pretty sure I saw on my bike ride here. And suffice it to say that this species is in numerous host-parasite networks. And so what I wanted to know was, do species that occur in multiple networks play similar roles across their different networks? And so to do this, I'll start with a sort of conceptual example of how you classify species into roles, and then discuss its sort of relevance to host-parasite networks. And so we can imagine a network of dots where there's links between them, but you can't see them for clarity, and they're colored based on the sort of community that they belong to. And so if we imagine this as a social network, different groups might be follow different interests or different age groups, where you have the people into martial arts here and the people into knitting up there, where each group corresponds to a different color. So there's 3 groups here. And there's a huge body of literature that has gone into basically the ability to detect these groups or modules in spatial or in ecological networks or networks generally. And so how we basically define species role is we first identify these communities, and then we look at the interactions of each species within its own community. So how good is a species, or how well connected is a species within its own little community, within its own sub-community, and how well does it link between different modules? And so this is like the captain of the football team that also is in chess club. Like he's able to sort of, or she is able to link different modules together. And so what we get from this is a 2-dimensional space that allows us to classify nodes as certain things, as fulfilling certain roles. And so on the right-hand panel, you can see along the x-axis, we have that among module connectivity, the ability of a species to link different modules, to link different groups. And on the y-axis, we have the within module connectivity, the tendency of a species to interact a lot within its own subcommunity. And these measures are standardized, which allows us to basically draw a threshold for each one of them. So we have a threshold for within module degree, and we have a threshold for among module connectivity that forms this, this sort of quadrats, where we can assign a species role based on where it lies in this 2-dimensional space. And we see on the bottom left those peripheral species, those that don't actually do that much interacting. And then we see, if we go sort of north of that, if we go up, we see when within module degree is high, we have what we call module hubs. And so these are guys that interact a lot within their own module, but not much outside of their own module. And then on the bottom right, we have connectors, which are basically ones that are not very good at interacting within their own community, but can connect different communities. And then lastly, we have the sort of jack of all trades, which are the network hubs. And this sort of framework has been applied previously to to plant pollinator networks by Pedro and colleagues. And one of the main findings from this was most species tend to be quite peripheral. They tend to not contribute very strongly, which is sort of a finding that is also seen other places in the network literature. Many species, a majority of species, do not contribute strongly to network structure. And so we wanted to look at this using the host-helminth data. And so we had data on about 300 locations and a total of right around like 40,000 species of hosts and parasites, so a solid chunk. And I had this great idea that I was going to make a map, and every location would be a pie chart, and it would show the sort of relative proportion of like connector species or module hubs. And I did it, and I wrote all the code for it, and I ran it, and I got this terrible plot that's just black circles distributed. And so what this means is that most species are peripheral. And so I analyzed each network and came up with something that looks like this, which is disappointing. But then if you look at it like this, it's slightly different, slightly less disappointing. And so this is all of the data plotted in that single, or in that two-dimensional space of the among module connectivity on the x-axis and the within module degree on the y-axis. And so you see the peripheral species at bottom left, but then you see there's a fair amount of module hubs. Like, things are doing stuff, basically. But this is all species and all locations mapped onto one two-dimensional space. And so basically each point here doesn't correspond to a species, it corresponds to an occurrence of a species in a location. And that's not what I'm interested in, because I want to see the conservation of species roles. And so I thought, and I'm still thinking, and this actually could not be the best thing, so if anybody has feedback as far as how to visualize this, I'd appreciate it. And so what I went with is we have not many of these network hubs, right? They make up less than 1% of the interactions, or of the plotted points. So let's just ignore them for a second. And then we have only 3 dimensions, and we can cut this down and look at it using ternary plots. And so basically these plots are able to visualize points in 3-dimensional space. And so we can look at the fraction of times for a given species that that species was classified as peripheral, a connector, or a module hub. And they're a little bit ugly, but a little bit cool as well. And so we have hosts on the left and parasites on the right, where each plotted point is a species. And what we still see many of the similar things as we did in that other plot, where you have a lot of peripheral species. And so points along that bottom bar are mostly peripheral species. They appear, especially like in this area. These are largely peripheral. However, we see these points here, which are actually saying like that's a good connector species. Yeah, it'll make more sense in a little bit, hopefully. And so this still doesn't really get at what I wanted to get at, because we don't really know. It does in a sense. And so points like right here are basically say that 33% of the time a species was classified as peripheral, 33% of the time it was classified as a connector, 33% of the time it was a module hub. That species role is not conserved by any means. But what I wanted to do was look at how species were classified if you considered all the interactions of all the parasites across all of space. And so this is actually a really common thing to do, especially with sort of macroecological studies, is to consider all hosts and all parasites regardless of location. Have they ever interacted? Yes or no, link them. And so what you're doing is creating a global network And so this is not the global network. This is actually less than 10% of the hosts and parasites that we had in the network. But imagine that it is global. And what we did is we calculated that same thing, the species roles, considering the global network, and then looked to see if the roles identified in the global network sort of matched up with what we saw in the local networks. And currently we see not really, right? And so each point now is colored by what that species was classified as in the global network. And if you squint hard enough at it, you start to see like a little more blue around here, which corresponds to species that are sometimes module hubs in local networks, and then module hubs in global networks. And you start to see a little more green here, and those are species that were identified as connectors in the global and tended to be identified as connectors in the local networks. But to look at it in a less confusing way, we can simply look at the number of times that a species was correctly identified in the local network to the global network. And so this is basically just a panel of histograms where each one of these would be what it was identified in the global network, and then the sort of number of species or the percentage of time that the species was correctly identified in the local network. And we see that especially for connector species and network hubs, we don't do a good job. Like the local role that a species plays oftentimes just doesn't fit with the role that a species plays in the global network. Module hubs, a little bit better. Like sometimes it's identified in the local network as a module hub, and it's also a module hub in the global network. And then peripheral species, which are the majority. There's so many peripheral species based on this analysis, actually had a really high percent match. But this is perhaps not very telling of anything. It's sort of if I walked up to someone at like a bar in Sevilla and said, "Are you drinking Cruz Campo?" And it's like, yeah, they're probably drinking Cruz Campo just because of the availability of Cruz Campo. So basically what I've learned from this so far is that species topological role, as identified here, isn't really conserved either for host species or for parasite species. However, this is ongoing, and I need to find a sort of better way to visualize it, and definitely. And so I'd appreciate any feedback on that. But to sort of wrap up what we've learned is the first question I looked at, how parasite communities and host communities change through space, and as well as network structure. And so I found that host and parasite communities become quickly dissimilar over even fairly short geographic distance, which goes along well with our understanding of sort of the ubiquity of distance decay relationships. But we saw that network structure did not follow this same pattern at all, which suggests that different species are actually playing functionally similar roles in these networks. And then we turned the question on its head a little bit and asked, do the same species play more similar roles? Like, are species roles conserved across space? And the main findings from this project, as they stand, is that the majority of species contribute very little to overall network structure. Also, it seems currently that the species roles in the local networks do not reflect what is happening in the global networks. And so the same species observed in multiple networks performed quite different roles. And so what does this really mean? It means that the context of the local network is incredibly important, right? And so to understand what role a species is playing in a local network, we can't look at other local networks in the area. It also means that the community likely really matters. And so the role in a species, or as a parasite, my role would differ in one network to the other as a function of the host community, right? And understanding this sort of spatial variation in species roles would allow a us to sort of quantify the effect of species invasions, because we can't presume that a species role in the native area would be the same as a species role in the invasive area, as well as sort of understand spatial and environmental gradients that could control species roles. So maybe host species play really strong roles where they're really abundant. That would make sense. Maybe parasite species play really strong roles when it's hot. So you should have environmental and spatial gradients that could relate to spatial roles. And sort of as I finish up, I did want to make a sort of plug for the funding that brought me here. And these are what's called HPC Europa grants. They give you access to high-performance computing systems, without which this work wouldn't be possible or would just take way too long. And so there's many, many high-performance computing centers distributed across Europe, and you are all eligible to apply for these grants, I believe. And there's a rolling deadline, and the next deadline is May 14th. And also, after I leave here and go back to Helsinki for a tiny bit, I will be heading back to the United States, and we'll be starting a professorship in theoretical ecology at Louisiana State University in Baton Rouge. And so anybody interested in becoming a grad student, a postdoc, or collaborating, this is my website. Feel free to contact me, talk to me, check it out. And with that, I'd like to thank you all for your attention, and I'm happy to take any questions, criticism. Yeah.